Hello, everyone, and welcome to today's webinar, Advances in Influenza Research, Developing Effective Vaccines. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sino Biological. To learn more, visit sinobiological.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Mohammed Safiarian, Associate Product Manager, Sino Biological. Dr. Safiarian, you may now begin your presentation.
Hello, everyone. My name is Mohammed Safiarian, and I'm Associate Product Manager at Sino Biological. Today, I would like to talk about the advances in flu vaccine development that results in the production of more effective vaccines. Here is an overview of today's talk. First, I begin by introducing the impact of influenza disease on public health and the importance of vaccination. Then I will briefly touch on the development and application of influenza vaccines. Then I will talk about the cytokine release in response to vaccination and the ways that vaccine efficacy is evaluated. Later, I will talk about the future trends and directions in influenza vaccine development. And finally, I will mention the services and products provided by Sino Biological to support the research and development of new vaccines against influenza. Influenza, commonly known as the flu, is a contagious respiratory illness caused by influenza viruses. Each year, influenza outbreaks pose a significant burden on public health worldwide. The flu can affect individuals of all ages, but certain groups, such as young children, the elderly, and pregnant women, or those with underlying conditions, are particularly vulnerable to severe complications. Influenza not only causes significant morbidity, but also puts a strain on healthcare systems, workplaces, and communities as a whole. Influenza is caused by influenza viruses, as we mentioned in the previous slide, which are classified into 4 types, A, B, C, and D. However, it's important to note that only influenza A and B viruses are responsible for seasonal epidemics that occur worldwide. The global impact of influenza is substantial. Each year, there are an estimated 3 to 5 million cases of severe illness caused by influenza, resulting in significant morbidity and mortality. It is worth mentioning that the burden of influenza is not evenly distributed among different populations. Certain groups, such as young children, older adults, pregnant women, and individuals with underlying health conditions, are at greater risk of developing severe complications. Accurate diagnosis, effective prevention, and appropriate treatment are key aspects in combating influenza. Let's explore these aspects together. Diagnosing influenza involves various methods, including antigen detection, virus isolation, RT-PCR, immunofluorescence assays, and rapid molecular assays. These diagnostic tools help healthcare professionals identify the presence of influenza viruses and determine the specific viral strains causing the infection. Accurate diagnosis is crucial for timely intervention and appropriate management. Prevention is undoubtedly the most effective approach to combat influenza. Vaccination remains the cornerstone of influenza prevention, and influenza vaccines are designed to stimulate the immune system to produce antibodies that recognize and neutralize the specific strains of the virus. In terms of treatment, most cases of influenza are managed symptomatically. Rest, hydration, and over-the-counter medications can help alleviate symptoms and support the body's natural healing process. However, antiviral medications can also be prescribed, particularly for individuals at higher risk of complications. The history of influenza is marked by several devastating pandemics that have had profound global impact. Since the infamous 1918 Spanish flu pandemic, there have been 3 more influenza pandemics: the Asian flu in 1957, the Hong Kong flu in 1968, and the most recent H1N1 pandemic in 2009. These pandemics result in significant illness, hospitalization, and unfortunately loss of lives. In addition to pandemics, influenza zoonotic events have occurred in the past 2 decades. These events involve the transmission of influenza viruses from animals to humans, posing a potential threat to public health. For example, avian flu H5N1, also known as the bird flu, and more recently, avian flu H7N9, have caused concerns due to their high mortality rates in humans. Influenza viruses have a unique structure. Let's explore key antigens and their functions in both diagnosis and vaccine development. Hemagglutinin, or HA, is a glycoprotein on the surface of the influenza virus. It plays a crucial role in viral entry into the host cells by binding to the receptors on the surface of the respiratory cells. It is a primary target for neutralizing antibodies induced by influenza vaccines as well as antiviral drugs. Neuraminidase, or NA, is another surface glycoprotein that facilitates the release of newly formed virus particles from the infected cells. It is the target of antiviral medications and also can be utilized in vaccine development. Nucleoprotein NP forms the backbone of the viral particle and is involved in the viral replication and packaging of the viral genome. NP is a conserved antigen and is most commonly utilized in diagnostic tests to detect the presence of influenza viruses. Matrix protein 2, or M2, is a transmembrane protein that functions in viral assembly and release. It is a target of antiviral drugs, and certain influenza vaccines contain M2 antigen to elicit immune responses. Non-structural protein 1, or NS1, is a multifunctional protein that helps the virus evade the host immune response. It is not a target for current influenza vaccines, but it plays a role in diagnostic tests to differentiate between influenza virus subtypes. Polymerase proteins, the influenza virus, has 3 polymerase proteins, PB1, PB2, and PA, that are essential for virus replication and transcription. These proteins are involved in the adaptation of influenza viruses to new hosts and play targets of new antiviral drugs. The development and application of influenza vaccines have played a pivotal role in our ongoing fight against this ever-changing viral infection. In the subsequent section, we will dive deeper into the fascinating world of influenza vaccine development, exploring the cutting-edge technologies, research breakthroughs, and future directions that hold promise for more effective, broadly protective influenza vaccines. The World Health Organization, or WHO, plays a critical role in monitoring and responding to the global threat of influenza. Through its Global Influenza Surveillance and Response System, or GISRS, the WHO closely monitors the spread and evolution of flu viruses worldwide. Based on its comprehensive surveillance data, the WHO, in collaboration with experts from various countries, makes annual recommendations for the strains to be included in the seasonal influenza vaccines. These recommendations, communicated through the WHO Global Influenza Updates and Consultation Meetings, guide the vaccine manufacturers in formulating the most appropriate vaccine composition for the upcoming flu season. For influenza vaccine production, 2 primary technologies are used: egg-based and recombinant methods. Let's compare the 2 technologies and examine their advantages and disadvantages. The egg-based vaccines have been the traditional method of influenza vaccine production for decades. They involve growing influenza viruses in chicken eggs, a process that has been well established and is relatively affordable. The egg-based approach allows for large-scale production of vaccines to meet the demand during the flu season. However, it does have some drawbacks. First of all, the production is time-consuming, and it takes approximately 8 months from the strain selection to vaccine availability. This lag time may pose challenges in rapidly responding to emerging antigens or pandemics. In addition, a subset of individuals may have allergy to egg proteins, limiting the suitability of egg-based vaccines for those individuals. On the other hand, recombinant flu vaccines represent a newer technology that does not rely on egg for production. Recombinant vaccines are developed using genetic engineering techniques, where specific proteins from the influenza virus are expressed in other cell types, such as mammalian cells or insect cells. This method offers several advantages. First of all, recombinant vaccines can be produced relatively quickly, taking around 5 months from a strain selection to vaccine availability. And moreover, since no eggs are involved in the production, recombinant vaccines are suitable for individuals with egg allergies. However, there are some drawbacks associated with the recombinant vaccines. For example, they can be more expensive to produce compared to egg-based vaccines, primarily because of the cost of the cell culture systems and the associated technology. And also, as a newer technology, there may be limited long-term efficacy data available for recombinant vaccines. Both approaches continue to be valuable in ensuring sufficient supply of influenza vaccines to protect populations worldwide. Let's look at the trivalent versus quadrivalent influenza vaccines. Another important consideration is the number of strains that has to be included in the vaccine. Two main options are available, trivalent and quadrivalent flu vaccines. Let's compare the two and look at the advantages and disadvantages. Trivalent flu vaccines traditionally contain 3 influenza strains, 2 influenza A and 1 influenza B strain. These vaccines have been in use for many years and have provided protection against the most prevalent strains. Over time, it has become evident that there are 2 distinct lineages of influenza B viruses that can circulate simultaneously. Trivalent vaccines, with their inclusion of only 1 influenza B, may not provide optimal protection against the other B strain. In contrast, quadrivalent flu vaccines contain 4 strains, 2 influenza A strains and 2 influenza B strains. By including both lineages of influenza B viruses, quadrivalent vaccines offer a more comprehensive coverage against circulating strains. Quadrivalent vaccines have gained significant momentum in recent years due to their ability to address the limitations of trivalent vaccines, and they're becoming increasingly available and widely used. There is a wide range of formulations and technologies employed to combat this ever-evolving viral infection, influenza that is. Let's explore some of the commonly used influenza vaccines and their characteristics. Number 1 is the live attenuated influenza vaccines, or LAIV, and this consists of weakened virus strains. While these strains can still infect and replicate within the body, they're designed to cause only mild or no symptoms. This type of vaccine is typically administered as a nasal spray and has been approved for use in certain age groups. The advantages include its single dose, it provides a strong and long-lasting immunity, and the disadvantages include it's not recommended for immunocompromised individuals. Second technology is inactivated influenza vaccines, which are produced by growing the virus in eggs and then inactivating them using chemicals. This process ensures that the virus is no longer capable of causing disease. Inactivated vaccines contain viral components such as hemagglutinin, neuraminidase proteins. Adjuvants, which are substances that enhance the immune response, are often added to improve the vaccine's effectiveness. The advantages of this type of vaccines is that they're safe for all ages, and the disadvantages include they require adjuvants and may need multiple doses. Third technology is virusomal vaccines that utilize reconstituted virus and envelopes that contain HA, NA, and lipids. These vaccines do not contain the entire virus, but instead focus on essential viral components. The virusomes facilitate the delivery of the components into the immune system and stimulate the immune response against the influenza virus. The advantage of this type is that they're safe, they provide good immune response, and the disadvantage is that they're expensive and they may need multiple doses. Split variant is the number 4 type of influenza vaccines, and they involve the inactivation of the virus followed by the opening or splitting the virus into its components. These components are then purified, ensuring that the only specific viral antigens such as HA or NA are present in the vaccine. Split variant vaccines induce an immune response against these viral components, providing protection against the corresponding influenza strain. Advantage of split variant vaccines include they provide good immune response, and the disadvantage is that they may not cover all the strains and they require adjuvants. Lastly, subunit vaccines, they utilize purified HA or NA proteins from the influenza virus. These proteins, which are the major antigens responsible for stimulating immune response, are combined with adjuvants to enhance the immune response. Subunit vaccines offer targeted protection against the specific strains of influenza. And the advantages of this type of vaccines is that they're safe, there is no longer a live virus, and disadvantages is that they may need multiple doses, and they provide a weaker immune response compared to others. Viral vector vaccines have emerged as a promising approach. These vaccines utilize specialized viral vectors to deliver genetic material encoding influenza surface antigens into the cells and trigger the immune response. Let's explore this fascinating technology in more details. There are 4 major steps in the function of viral vector vaccines. The first step is in viral vector influenza vaccine development involves the insertion of the genetic material that codes for influenza surface antigen into the replication defective viral vector. One commonly used viral vector is the adenovirus, which is modified to be capable of replicating and causing disease. In the second step, the modified adenovirus vector carrying the genetic material encoding influenza surface antigen is introduced into the cells, either through injection or other delivery method. Once inside the cell, genetic material is released, and the cells begin to produce the influenza surface antigen. In the third step, the immune system recognizes the newly produced influenza surface antigen, and this recognition triggers an immune response where immune cells such as T cells and B cells are activated. The immune response aims to eliminate the cells displaying the influenza surface antigen and generate an immunological memory against the virus. Finally, the immune system becomes primed and ready to recognize and attack the influenza virus upon subsequent exposure. Various viral vectors have been harnessed to deliver genetic material encoding influenza antigens. Let's explore some of the major viral vectors used in the development of these vaccines, along with their advantages and disadvantages. First group are adenoviruses, as mentioned previously. Adenovirus-based vectors exhibit high immunogenicity and have been extensively used in clinical trials for influenza vaccine development. They have the ability to induce strong immune responses without the need for additional adjuvants. One drawback of adenoviral vectors is pre-existing immunity in individuals due to previous exposure to adenoviruses, which can affect the immunization efficacy. Second group are arenaviruses. Arenavirus-based vectors offer high immunogenicity and are capable of eliciting robust CD8+ T-cell responses, which are crucial for antiviral defense. They can evade neutralizing antibodies, enhancing their potential as effective vaccine carriers. One potential drawback of arenavirus vectors is their potential to cause neurological problems, which necessitates careful consideration during their development and use. Third group are Newcastle disease virus. Newcastle disease virus-based vectors have the capacity to induce mucosal, humoral, and cellular immune responses, making them attractive candidates for influenza vaccine development. They're highly proliferative in chicken embryos, allowing for large-scale immunization efforts. Similar to other viral vectors, pre-existing immunity against the Newcastle disease virus can affect the efficacy of immunization. Fourth group are baculoviruses. Baculovirus vectors offer a high level of recombinant protein expression, making them valuable tools for generating large quantities of influenza antigens. And finally, fifth group are herpes viruses. Herpes virus-based vectors display a high immunogenicity and are known to be very host-specific, enhancing their potential as vaccine carriers. They can accommodate the expression of large sequences up to 10 kilobase pairs, allowing for the incorporation of multiple antigens. Competition between vector proteins and expressed foreign antigens for immune response can be a potential drawback of herpes virus vectors. As we strive to ensure the safety and efficacy of influenza vaccines, monitoring the immune response they elicit is of paramount importance. One crucial aspect of this monitoring is examining cytokine release in response to vaccination. Cytokines are signaling molecules produced by immune cells that play vital roles in coordinating and regulating immune response. When we receive an influenza vaccine, it stimulates our immune system to mount a protective response against the virus. This immune response involves the production of various cytokines that serve as messengers orchestrating activities of immune cells. So monitoring the cytokine release following the influenza vaccines provide valuable insights into the immune system reaction to the vaccine. Cytokine profiling can help identify patterns of cytokine production associated with predictive immune responses, providing valuable information for vaccine development and formulation. As mentioned previously, monitoring cytokines is an essential component of ensuring vaccine safety and evaluating the immune response elicited by vaccines. Cytokine levels provide valuable insights into the type and strength of immune response triggered by vaccines. Certain cytokines, including interleukin-1 and tumor necrosis factor alpha, have the ability to activate immune cells like macrophages and dendritic cells. These cells play a critical role in presenting the vaccine antigens to other immune systems, initiating a cascade of immune reactions. Additionally, cytokines such as interferon gamma and interleukin-2 help activate T cells, which are essential for recognizing and attacking infected cells. The release of cytokines in response to vaccination is a normal and necessary part of immune response. It primes the immune system, ensuring that it's prepared to recognize and respond to specific pathogens targeted by the vaccines. However, it's important to note that the excessive or prolonged cytokine release can sometimes lead to harmful inflammatory responses. This is why monitoring cytokine level is a critical aspect of vaccine development. By carefully assessing cytokine profiles, researchers can gain insight into immune responses, effectiveness, and safety. This allows us to identify any potential imbalances or adverse reactions, enabling further investigation and refinement of vaccine formulations. Excessive cytokine production, if left unchecked, can lead to serious adverse events, including a condition known as cytokine storm syndrome. Cytokine storm syndrome involves an uncontrolled and overwhelming release of cytokines, resulting in a hyperactive immune response that can be detrimental to the body. Therefore, closely monitoring cytokine release is of utmost importance in vaccine development and evaluation to ensure the safety and efficacy of the vaccines. As mentioned in the previous slide, monitoring the level of specific cytokines can serve as a valuable marker for assessing vaccine safety. In this section, we will explore some key cytokines and their roles as biomarkers of vaccine efficacy. Laboratory assays such as ELISA are commonly used to measure cytokine levels in blood or other biological samples. These assays using antibodies against the cytokines can detect the presence and concentration of specific antibodies, such as interleukin-2, interleukin-4, interleukin-6, interferon gamma, and tumor necrosis factor alpha. For example, increased IL-2 levels following vaccination can indicate successful T cell activation, suggesting a robust cellular immune response. Elevated IL-4 can indicate effective B cell activation, implying a potent antibody response following vaccination. Monitoring IL-6 level can provide insight into the activation of both cellular and humoral immune pathways. Increased G-CSF following vaccination can suggest highlighted neutrophil production, enhancing the immune system's ability to combat infection. Monitoring TNF-alpha and IL-1-alpha can provide insights into the activation status of these antigen-presenting cells, indicating successful immune priming. And increased levels of interferon beta and interferon gamma following vaccination can suggest the induction of a robust cell-mediated immune response. As we continue to combat influenza and strive for more effective prevention strategies, ongoing research and innovation in vaccine development are paving the way for future advancements. In this section, we will explore some exciting directions for the future of influenza vaccine development. In the continuous battle against influenza, scientists and researchers are constantly exploring innovative approaches to improve the selection of seasonal flu strains for vaccine development. Traditional serological methods, which involve testing blood samples for the presence of antibodies, can be labor-intensive and inconsistent. However, with advancements in bioinformatics and computational tools, we now have new avenues to anticipate and track the natural evolution of flu viruses. One such tool is NextFlu, an online platform that utilizes bioinformatics to visualize the genetic evolution of seasonal flu viruses. By analyzing the genetic sequences of flu viruses obtained from surveillance data, NextFlu can provide insights into how these viruses are evolving and changing over time. This information is crucial for predicting the most likely strains to circulate in upcoming flu seasons, allowing researchers to make informed decisions regarding vaccine composition. Another valuable tool in the selection of seasonal flu strains is antigenic cartography. This tool utilizes the surveillance data provided by the World Health Organization to analyze the antigenic properties of flu viruses. By plotting the antigenic relationships between different strains on a map, researchers can identify clusters of viruses with similar antigenic characteristics. This information helps understanding how flu viruses evolve and how they may respond to immune pressure, guiding the selection of strains for vaccine formulation. By targeting these viral components, including HA stalk domain, NP, and NA, researchers are striving to develop a universal flu shot that provides broad and durable protection against multiple influenza strains. These approaches focus on conserved regions of the virus which have the potential to induce immune responses capable of recognizing and neutralizing a wide range of influenza viruses. One key viral target is HA stalk domain. This region of the HA protein plays a crucial role in cell membrane fusion, allowing the virus to enter the host cell. Compared to the highly variable head domain, the stem domain of the HA stalk is relatively conserved across different influenza strains. By targeting the HA stalk domain, researchers aim to induce immune responses that can neutralize a wide range of influenza viruses. Another promising target is matrix protein 2, or M2. M2 is highly conserved among different influenza strains, making it an attractive target for universal vaccines. The nucleoprotein, or NP, of the influenza virus is also another attractive target for a universal vaccine. NP is involved in viral replication and is highly conserved across different strains. Additionally, the neuraminidase, or NA protein of the influenza virus, has been identified as a potential target for universal vaccine development. In the quest to develop a universal flu vaccine that can provide broad and long-lasting protection against diverse influenza strains, scientists have explored various platforms that offer innovative approaches to vaccine development. Here are some of the examples. One platform is the virus-like particle. It involves the co-expression of the HA and NA and M genes together, which generate VLPs resembling the structure of the influenza virus. VLPs mimic the native virus without the genetic material, making them safe for use in vaccine. By presenting the viral antigens in a highly organized and immunogenic manner, VLP-based vaccines can elicit strong immune responses. Another platform is nanoparticles. In this approach, the ferritin protein and HA of the influenza virus self-assemble into nanoparticles. These nanoparticles display the viral antigens in a highly ordered and repetitive manner, enhancing their immunogenicity. By leveraging the self-assembly property of ferritin and HA, nanoparticle-based vaccines have the potential to induce the robust and cross-reactive immune responses. The viral vector platform utilizes adenoviral vector as delivery vehicles for the required antigen. Adenoviral genomic sequences are engineered to include a gene cassette encoding the desired viral antigens. When administered, the adenoviral vector delivers the antigenic genes into the host cell, stimulating the immune system to generate immune response. Nucleic acid-based platforms such as DNA and mRNA vaccines have also shown promise in universal flu vaccine development. In the DNA vaccine approach, a plasmid carrying the gene cassette encoding the desired immunogen is constructed. The plasmid is then delivered into the host cell, allowing them to produce the viral antigen and trigger immune response. Similarly, in mRNA vaccines, mRNA molecules representing the immunogen are formulated and delivered into the host cell. Once inside the cell, the mRNA instructs the production of the viral antigen and initiating the immune response. The pursuit of a universal influenza vaccine has gained significant momentum. These vaccines candidates utilize various platforms, including VLPs, nanoparticles, viral vectors, and nucleic acid, as previously mentioned. Several of these vaccines candidates are currently in various phases of clinical trials, including phases 1 through 2. Let's explore some of the key hurdles faced in the pursuit of a universal influenza vaccines. One major challenge lies in the evaluation of the novel influenza vaccines during preclinical trials. Unlike seasonal influenza vaccines, which can rely on established endpoints such as antibody titers and seroconversion rates, universal vaccines target broader immune responses that go beyond strain-specific antibodies. Currently, there is a lack of uniform evaluation criteria for these novel vaccine candidates, making it challenging to compare their efficacy across different studies. Another obstacle is in the development of universal vaccines is the absence of established correlates of protection for broadly cross-reactive immune responses. Traditional measures of vaccine efficacy, such as neutralizing antibody titers, may not fully capture the effectiveness of a universal vaccine that aims to provide broad protection against diverse influenza strains. Furthermore, the funding landscape poses a significant challenge. Despite the potential impact and importance of universal influenza vaccines, funding for their development is limited. Resources are often focused on seasonal influenza vaccine production and distribution, leaving a funding gap for research and development of universal vaccine strategies. At Sino Biological, we are dedicated to supporting and advancing the field of influenza vaccine research. As a globally leading biotechnology company, we offer a wide range of high-quality bioreagents that are instrumental in the development of influenza vaccines. In this section, we will dive deeper into the specific ways in which Sino Biological supports influenza vaccine research. We take immense pride in offering an extensive range of high-quality bioreagents that are instrumental in advancing influenza research worldwide. Our comprehensive product portfolio includes a remarkable selection of influenza antigens, subtypes, strains, proteins, antibodies, genes, and ELISA kits. With over 10 influenza antigens, 60 subtypes, 375 strains, more than 650 proteins, more than 200 antibodies, and more than 1,500 genes and ELISA kits, our collection provides an unparalleled resource for scientists and researchers in the field. With our ProVir viral antigen collection, researchers gain access to a wide range of antigens that accurately represent the strains recommended by the World Health Organization for the past decade. This collection includes HA, NA, NP proteins, which are key targets for vaccine-induced immune responses. By studying the interactions between these antigens and antibodies generated by vaccination, researchers can gain insight into the effectiveness and potential limitations of influenza vaccines. Additionally, our ProVir viral antigen collection is complemented by a diverse selection of monoclonal antibodies. These monoclonal antibodies serve as indispensable tools for assay development, allowing researchers to design and optimize assays that precisely measure antibody responses against the specific viral antigens. By utilizing Sino Biological's ProVir viral antigen collection, researchers can investigate and characterize the immune response induced by influenza vaccines. paving the way for the development of more effective preventive strategies. Sino Biological's broad-spectrum influenza antibodies offer a comprehensive solution by targeting conserved regions of the virus that are shared among multiple strains, including both influenza A and B. These antibodies are used in diagnosis of various influenza subtypes, which, with their ability to react with most flu strains, they offer an efficient and reliable means of detecting the presence of influenza viruses in clinical samples. Allow me to highlight some of the notable catalog antibodies included in our broad-spectrum antibody collection. Anti-influenza A HA antibody. This antibody specifically targets the HA protein of influenza A virus, including subtypes H1N1 and H3N2. By recognizing the HA antigen, it facilitates the detection and analysis of influenza A strains. Anti-influenza BHA antibody, anti-influenza A NP antibody, and anti-influenza B NP antibody. Likewise, they're designed to specifically recognize their respective antigens of influenza A or B viruses. These catalog antibodies exemplify Sino Biological's commitment to providing high-quality reagents for influenza research and diagnosis. With their broad spectrum of activity, they offer researchers and healthcare professionals a powerful tool set to accurately detect and monitor influenza infections. We are delighted to present Sino Biological's comprehensive range of ELISA kits designed for the detection of cytokines. These kits play a pivotal role in gaining valuable insights into the specific immune responses elicited by vaccines during the development and evaluation of influenza vaccines. As mentioned previously, measurement of cytokines such as TNF-alpha, IL-6, HGF, and others provide a deeper understanding of the immune response provided by the vaccine. These cytokines act as signaling molecules, and they're very important because if they left unchecked, they might cause a cytokine storm. By accurately quantifying their levels, we can assess the effectiveness and safety of the influenza vaccines. Our ELISA kits cover a wide range of essential cytokines involved in the immune response, including IL-1A, IL-2, IL-4, IL-5, IL-6, TNF-alpha, and interferon gamma, G-CSF, NGF, and so on and so forth. In addition to the individual ELISA kits, Sino Biological also provides ELISA pair sets for several cytokines, including IL-1A, IL-5, IL-6, IL-8, and so on. These pair sets offer a comprehensive solution by providing both capture and detection antibodies, ensuring optimal performance and sensitivity in cytokine measurement. ProVer, as mentioned previously, stands as a testament to our commitment to providing the scientific community with a comprehensive and high-quality viral antigens. With over 1,200 products encompassing more than 90 virus types and subtypes, and more than 400 viral strains, this extensive collection offers an unparalleled resource for studying a wide range of viral pathogens. Our ProVer collection encompasses a diverse array of viral antigen, enabling researchers to investigate the intricacies of viral infections and host immune responses. With an extensive catalog, we offer over 8,000 proteins, 14,000 antibodies, 45,000 genes, and more than 600 ELISA kits. We provide a diverse selection of tools to support your research needs. Our vast collection encompasses various species, including human, mouse, rat, and more. From recombinant proteins to monoclonal antibodies, our high-quality products undergo rigorous testing and validation to ensure reliability and consistency. With our extensive portfolio, we aim to provide researchers with the necessary tools to advance scientific discovery and drive innovation in the field. Our CRO services are designed to support your research and development needs across various fields, including influenza vaccine research. We offer a range of services, including recombinant protein expression, recombinant antibody production, monoclonal antibody development, and polyclonal antibody development. As a global company, our reach extends to over 190 countries, and we have established a strong presence worldwide. We are committed to delivering high-quality products and services that meet the needs of researchers and scientists around the globe. Our commitment to quality is evident in our ISO certificates. These certifications reflect our dedication to maintaining rigorous quality management systems to ensure the consistency and reliability of our products and services. We are proud to see our products cited in over 10,000 publications as of September 2022, appearing in prestigious and high-impact journals. This recognition demonstrates the trust and confidence researchers have placed in our reagents and services. At Sino Biological, we have established a strong global presence with our headquarters located in Beijing, China. In addition to our main office, we have strategically placed branches in various cities to better serve our customers. Our branches can be found in Guangzhou, Shanghai, Taizhou, Suzhou within the China, allowing us to cater to the diverse needs of researchers and scientists across the country. Furthermore, we have expanded our reach internationally with branches in Tokyo, Frankfurt, Pennsylvania, and Texas. These locations enable us to provide efficient and reliable support to our customers worldwide, ensuring prompt delivery of high-quality bioreagents and comprehensive CRO services. With our global network, we are dedicated to facilitating scientific advancements and collaborations across the borders. And on that note, we would like to express our sincere gratitude for your time and attention today. At Sino Biological, we're dedicated to advancing scientific research and contributing to the global fight against infectious diseases. We're proud to provide a wide range of high-quality bioreagents, including antigens, antibodies, ELISA kits, and more, to support influenza vaccine development and other critical areas of research. If you have any further questions or require additional information, or you would like to explore our offerings in more detail, please don't hesitate to reach out to our team. We're here to assist you and provide tailored solutions to meet your research needs. Thank you once again for your participation and interest.
Thank you, Dr. Safarian, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how do different influenza strains emerge and spread globally?
Well, influenza strains can emerge through genetic mutations and reassortment events, which allows them to evade existing immunity and spread rapidly. So mutations in influenza virus's genetic material can occur over time, which leads to the development of new strains. But for reassortment events to happen, two different influenza viruses usually infect the same host and exchange genetic material, and that results in creating new strains. And these emerging strains, particularly those with the potential to infect humans, can then spread globally through human-to-human transmission, which is facilitated usually by international travel.
Thank you. Our next question here is, what are the primary methods used to evaluate the effectiveness of influenza vaccines?
Well, just like many other new drugs, the effectiveness of influenza vaccines is usually evaluated with randomized controlled clinical trials, which involves vaccinated individuals and control groups. Researchers and companies, they monitor the reduction in laboratory confirmed influenza cases among vaccinated individuals and then compare them to unvaccinated individuals. They also evaluate the severity and duration of flu symptoms, hospitalization, and overall vaccine safety between the two groups. And in addition, serological studies measure the levels of influenza-specific antibodies in vaccinated individuals to evaluate immune responses and to correlate them with the protection against the virus.
Wonderful, thank you. Our next question here is, are there any other measures in addition to vaccination that can help prevent influenza infection?
Yes, absolutely. So vaccination is the most effective way to get immunity against influenza. But in addition to vaccination, adopting good respiratory hygiene practices can prevent the transmission of the influenza. These practices usually include regular hand washing with soap and water or using hand sanitizers. covering the mouth and nose when coughing or sneezing with a tissue or the elbow, and properly disposing of the used tissues. It's also important to avoid close contact with infected individuals, especially during flu outbreaks. Practicing a healthy lifestyle, including getting enough sleep, maintaining a balanced diet, staying physically active, and managing stress can also contribute to maintaining a strong immune system, which can help in preventing influenza and basically any other infection.
Wonderful, thank you. Our next question here is, what makes Sino Biological's ProVir collection unique and valuable for infectious disease research and vaccine development?
Well, Sino Biological's ProVir collection is a very valuable resource for infectious disease research and vaccine development because of its extensive coverage and high quality standards. So with over 1,200 antigens encompassing more than 90 virus types and subtypes and more than 400 viral strains, our ProVir collection is the world's largest recombinant viral antigen bank. So this extensive coverage allows researchers and companies and vaccine developers to access a wide range of viral antigens for studying immune responses, basically developing diagnostic assays and evaluating vaccine candidates. What sets us apart is that the ProVir collection is of very high quality standards. So Sino Biological has several ISO certifications, which ensures our customers that the antigens are produced following rigorous quality control processes, and the availability of full product data, including batch-specific information, further enhances the reliability and reproducibility of the experiments that are done using ProVir antigens.
Excellent, thank you. What is the role of adjuvants in influenza vaccines?
Well, adjuvants are substances that are usually added to some types of vaccines to enhance the immune response. In influenza vaccines, adjuvants help boost the body's immune response to the vaccine by stimulating the immune system, and this results in a stronger and more effective immune response against the influenza virus. Adjuvants basically work by activating certain cells and signaling pathways in the immune system, which help to enhance the recognition and processing of the viral antigens that is present in the vaccine. And this leads to a more robust immune response, including the production of specific antibodies that can neutralize the virus, and also T cell responses that provide cellular immunity. In case of influenza vaccines, adjuvants again can improve the magnitude and duration of the immune response by increasing the production of antibodies and also activating other components of the immune system.
Perfect, thank you. Another question we have here is, how do cytokines play a role in the immune response to influenza vaccines?
Well, cytokines such as TNF-alpha, interleukin-6, interferon gamma, they're released as part of the immune response to influenza vaccines. And these small proteins act as signaling molecules that regulate and coordinate various immune responses. Cytokines help activate immune cells, such as T cells and B cells, which are crucial for mounting an effective immune response against the influenza virus. They also stimulate the production of antibodies, enhance the recruitment of immune cells to the site of infection, and contribute to the overall immune defense against the virus. Here I should also point out that the Sino Biological offers a wide range of products to monitor the release of cytokines and to ensure the vaccine safety. These products include ELISA kits for the detection of various cytokines, which are used to measure the immune response generated by the vaccine during the development and evaluation. In addition, Sino Biological offers a variety of validated antibodies developed against cytokines, and these antibodies are used again to detect and measure specific cytokines.
Thank you. It looks like we have time for one more question here. What is the nucleoprotein antibody, or I'm sorry, why is the nucleoprotein antigen mainly utilized for diagnostic purposes among the three main influenza antigens?
Well, the nucleoprotein, or NP in short, is often used for diagnosis, as you mentioned, and this is because the NP is highly conserved among different influenza virus strains, which makes it a reliable target for detecting and identifying influenza infections. Antibodies specifically targeting the NP antigen can be used in diagnostic assays such as ELISA or rapid diagnostic tests to detect the presence of influenza viruses in patient samples. This allows for broad detection across different influenza strains and basically facilitates the timely identification of the influenza infection. So you basically can tell in a very short time that someone has influenza or not, which is very important in patient management and public health surveillance efforts. I would also like to mention that Sino Biological offers a large selection of antibodies against influenza. Some of the antibodies offered by Sino Biological include anti-influenza AHA, anti-influenza BHA antigen, anti-influenza A NP antigen, and anti-influenza B NP antigen. As mentioned during the talk, these antibodies are essential tools for researchers involved in the influenza studies, companies that are developing diagnostic assays, and those who are working in the field of vaccine development.
Thank you again, Dr. Safarian, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sino Biological, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.